All right, diving in today. Bead milling. You listeners sent in a bunch of interesting stuff on this. Sounds pretty intense. Basically, a way to crack open cells, right?
Yeah, pretty much. It's all about getting to the good stuff locked up inside those cells, the proteins, enzymes, all that. And bead milling, well, it's a pretty neat way to do it.
So our mission today is to really get this. How does it actually work? When is it the best tool for the job? What are the downsides? Your sources are calling it a physical method. Mechanical force and these tiny beads. That's it. No harsh chemicals involved.
Exactly. It's all about brute force at a microscopic level.
Yeah.
And yeah, that keeps things cleaner, which is a huge plus. When you're working with delicate molecules, you don't want to mess up.
Okay, I'm looking at some diagrams here.
Yeah.
So we've got these beads colliding with cells. Talk about a rough day for those cells.
Yeah.
Shear forces, impaction, attrition, like a microscopic demolition derby.
Exactly. It's controlled chaos in there. You've got your cell suspension, mixing the beads, and then bam, agitate, agitate, agitate. Shear is that tearing action, impaction is the direct hit, and attrition is that constant grinding. Imagine tiny wrecking balls going to town.
I bet those little cells don't stand a chance.
Yeah.
So picture this. I'm in the lab, and I need to bust open some cells. There's probably test tubes on a shaker going crazy, right?
Yeah, you got it. For smaller samples, batch bead mills are the go-to. Like you said, tubes on a shaker, maybe a vortex mixer. But for bigger jobs, like in industrial settings where you're processing tons of material, they use continuous bead mills, constantly feeding the cell mix and beads into this chamber with a seriously powerful agitator.
So continuous means non-stop cell crushing.
You got it. But there's a catch-all that friction means heat and lots of it, which is why those continuous mills have built-in cooling systems. Keeping things cool is crucial.
Makes sense. You don't want to cook those precious molecules you're trying to extract. Now, the materials mention bead size being important. Smaller beads for smaller cells. And different types of beads, too. Glass, ceramic, even zirconia.
Right. It's all about choosing the right tool for the job. Smaller beads can get into tighter spaces like those tiny bacteria. And the material, well, that matters too. Zirconia, that's a tough one. Perfect for really robust cell walls, like in yeast.
So like using a sledgehammer instead of a regular hammer, depending on what you're trying to break.
Pretty much. Then you've got bead loading, that's the ratio of beads to cells. More beads, more collisions, more disruption. But too many beads and things can get, well, thick, like trying to stir concrete.
And of course, the faster you agitate, the more those beads are bouncing around, the faster you break those cells open.
Right.
But again, heat. Same with processing time. Longer processing, more disruption. But you got to watch that temperature. Seems like a lot of factors to balance.
Absolutely. It's about optimization, finding the sweet spot. For example, if you need longer processing times to really pulverize those cells, batch mills will often use cycles, agitation, then cooling, like giving those little molecules a break from the heat.
Clever. So I'm seeing bead milling being used on all kinds of cells bacteria,
Yep.
including the tough ones like mycobacteria, yeast, fungi, algae, plant cells. What's the common thread? Is it always about getting those intracellular components? Enzymes, proteins, and stuff from the cell wall?
Yeah, essentially. Think about it whether you're a researcher trying to understand how a specific enzyme works or a company manufacturing a protein-based drug. It all starts with getting inside those cells. And that's where bead milling shines. It's scalable, adaptable, and incredibly effective.
All right, so it sounds like a pretty amazing technique,
Yeah.
but I'm guessing there are some downsides. You already mentioned heat, and I imagine it's not exactly quiet either. Plus, once you've busted open all those cells, those tiny beads must be a real pain to separate from the rest of the stuff, right?
Good points. The noise can be a factor, especially in those large-scale operations. And yeah, separating those nanometer-sized beads from the lysate, which is what we call the broken cell soup, that can be tricky and require special techniques. And of course, there's the upfront cost. Those bead mills aren't cheap. Plus, even though it's just physical force, you still have to be careful. Those forces can damage sensitive proteins if you don't keep that temperature in check.
So the key takeaway here, bead milling is a powerful and versatile tool, especially for tough cells, to get at all the valuable stuff inside. But you got to be really careful. Controlling those operational parameters, especially temperature, is key to getting the best results.
Exactly. It's about finding that balance, maximum cell disruption, while keeping those precious molecules intact.
This has been a fascinating deep dive into bead milling. Makes you wonder what other seemingly simple physical processes might be happening in biological systems, things we haven't even discovered yet. Something to ponder. Until next time.